Hello, everybody. It's April 10th, 2021. You are at the place to be. This is the most fun you can have at 9 p.m. Eastern Time on a Saturday night because we stretch our brains and we stretch our vision into the future. You might ask why we do this stuff. It's because when we do research, it literally amazes us. And it's like ever so relevant to the world that we live in. And of course, if you've been following us episode by episode, which is beneficial to do because they kind of add on to each other, that the advances in the connection between the human body and, of course, experimental lab animals as well, and computers, and the software side of it, where they can map out all sorts of more and more intricate parts of the genes in our bodies and in our cells, and right down to the proteins and how to address, tag, and be able to influence and control them is absolutely amazing. So we do this because it's so exciting, and we want to bring that alive to you. We want to bring the excitement to you as well. Going to just, before we go too much further here, we're going to just read to you as quickly as I possibly can our community guidelines disclaimer. And that is this: the points of view and purpose of this video is not to induce fear or incite any form of violence or harass or bully in any way, but rather to share and exchange thoughts with other like-minded individuals curious about the subject at hand, to encourage conversation and awareness. The opinions expressed in this video belong solely to the persons espousing them and are not a representation of the channel cyberhumanfrontiers. None of our videos claim to give medical, legal, political, or financial advice, and this content is created entirely for your entertainment. We may from time to time incorporate certain copyrighted works not specifically authorized to be used by the copyright holder, but which we believe are relevant and in good faith are protected by federal law and the Fair Use Doctrine for one of the more reasons noted in the Copyright Act of 1976. That being said, if you've been with us, you know that we've been delving into nanotechnology, nano-sized particles, and optogenetics in the last couple of episodes. And we're going to go a little bit further with that now, but we're going to kind of go back and forth with some content produced by some other people, which is both some of it's a little bit lighthearted and some of it's a little bit deeper, and we're going to kind of weave that together with what TMC will bring into this, which will be our actual substantiation with patents and devices and what's at the cutting edge of the field as industry and academia are developing and refining it. That being said, we are just going to check audio with TMC. He had a minor computer glitch going on, so TMC, just checking in with you. What are you sounding like?
I think it's fine. I hear you now just fine. So
perfect. That's excellent. All right. Do you have anything to add to that initial little blurb, or shall I get right into something that I'd like to start us off with?
No, I think you should proceed. I'll do what I can to dig up things on the fly, but we'll see where this goes. Yeah,
I'm sure. And so you kind of know where it's at. All the links are in your your Discord. I sent them to you.
Very good.
Okay, so we are going to queue up. Ooh, let me close this other window here. Okay. All right. So testing 1, 2. Yep. Looking good. You guys out in the chat. Glad to see you there. And I'm going to try to entertain. So this is, and by the way, the links to all of these little productions that we will show you are in the underneath our video. And you can certainly go ahead and check those out further. It's all really cool stuff. So the title of this is Scientists Putting the Brain of a Worm into a Robot. And if you know how this goes, we always usually start simply and then move toward ever more complex applications once we get a clue how things work. So we start off with modest beginnings, merely trying to put the brain of a worm into a robot. But if you put on your science fiction thinking cap and you look forward to where that could be in about 10 years or 50 years or 2 years, then you can guess what, another, like a bigger milestone might be. So I want to thank these content producers for doing the fine work that they do, and we're going to let this one fly. Let me just make sure that the audio is going to work right. TMC, if there is any issue with it, just flag me, because I won't be able to tell. And anybody out in the chat, let me know as well. Okay.
This looks like a robot being controlled by an Ewok, but actually the robot isn't being controlled at all. What you're looking at is a copy of the brain of a Caenorhabditis elegans, or C. elegans. This is the digital brain of a worm on a computer chip inside a Lego robot. This little guy is a simulated brain that's just navigating on its own. Well, navigating the best way.
Okay, so here's what they did. They figured out how the brain of the worm works, and they mapped out every little part of it, and then they digitized it, basically. They took all of those connections, all of those neuron connections in the worm's brain, put them on a chip, and stuck it in a robot. He's going to tell you more.
That a worm bot can. To do this, the scientists put sensors on the robot's nose and tail. So if they meet a wall, they know to turn around, just like a real C. elegans. But as simple as this robot seems to be, getting to this point took a long time. Researchers spent decades looking at each and every cell in the C. elegans and how it works with the cells around it. Then other researchers built a program that could mimic.
All right, so check out this picture. I'm only guessing, but I'm pretty sure that every one of these little blocks in this matrix that you're seeing on your screen represents a neuron, and that each one of those blocks has got programmed in it almost like a macro in an Excel spreadsheet, or almost like a linkage, a hyperlink connected between one and another component in a matrix in HTML. That these things are in a sort of a wiring. You're not seeing the wiring behind the scenes, but I'm sure that they've mapped out those interconnections between them all too. And what you're seeing here might be showing the activity of them as that siliconized worm brain thinks, if you will.
That interaction. Each one of these boxes represents a neuron interacting with the environment. They could do this because scientists have a complete map of every single cell in the C. elegans and their functions. It's the worm's connectome, and it holds around 1,000.
So think about that. They've mapped out every single cell in that worm's brain. And it's not a big worm. It's as worms go, this is a fairly small worm, and that's the way you start off. Map out all the neurons in a 1,000 cell worm, and then build up from there. Kind of like what we did with computers. In the 1980s, we had computers that only had a couple 100,000 transistors on the chip. If you remember old 486s. Now there are over 1 million transistors on it. In fact, well more than that. I got to look it up. It's a lot. And of course, the sophistication carries on. So this is where it starts, and that's where it goes.
1,000 cells and all 302 neurons of the worm. So once you've digitized how every cell and neuron interact, you sort of have the digital version of the C. elegans. By simulating a brain at the cellular level, the researchers can watch larger aspects like movement, for example, emerge naturally. Or, put another way, they turned on the simulation and let the worm bot just be a worm bot.
So that's fascinating. I think he said 302 neurons. Well, that's really a good way to start. The human brain is, it's trillions of neurons, all connected and interconnected in one way or another. So this is indeed starting very simple. And all it can do is control a kind of a simple robot that can go back, forward, and steer left and right. So it's some place to start. but it's a very interesting direction to be moving in as it becomes more powerful. And by the way, in previous episodes, we've covered some of the more powerful applications of the inroads into the understanding of human brain neurology have already brought us. But I thought this was kind of entertaining.
They do this by taking the connectome and some algorithms and a precise anatomical map and combining them together. Then they throw that into a simulator, which is very complicated, and I don't pretend to understand how it all works. But the important thing is what happened once they flipped on the power. The robot just behaved like a living C. elegans. I mean, almost. The researchers are starting small. They didn't want the robot to do a lot of stuff, so it's just interacting with the muscles of the robot. It doesn't have independent instinct, it's not going to start looking for food or anything yet. Those behaviors are on the agenda for the researchers, but they just wanted to get the digital organism moving first. Now, I know what you're thinking. This is a huge step towards some crazy...
Digital organism. Okay.
These scientists digitizing and uploading human brains into robots. Not really. Not really. These scientists are mapping this worm's brain and creating an accurate digital simulation because we still don't know a lot about how our brain works. This project's success could completely change the way we map and understand our own brain. Plus, with this application, we could possibly get rid of squishy living organisms in labs and could rely...
Get rid of squishy living organisms in labs. Back that up again. That sounded good because they're squishy, but they're living too. Living is so messy.
Plus, not really. Not really. These scientists are mapping this worm's brain and creating an accurate digital simulation because we still don't know a lot about how our brain works. This project's success could completely change the way we map and understand our own brain. Plus, with this application, we could possibly get rid of squishy living organisms in labs and could rely on computer simulations of those organisms instead. There's nothing to fear. C. elegans are only about 1 millimeter long. It'll take a...
So I'm kind of a squishy organism. Wouldn't it be neater and cleaner if I mean, what if I didn't have to be so squishy and I could be reliable, electronics, I wouldn't get tired. Maybe there are some advantages to that. So granted, it's going to take a lot more than mapping out just 302 neurons to be able to encompass all of my eccentricities, but it's some place to start. And I don't know, is that something that we should be hoping for? Maybe, maybe not. Seems like nature did a really good job on building organisms that function the way they do, but there's always the next thing. And that's pretty much all he has to say here. I thought it was just kind of entertaining and fascinating.
While before we start digitizing your brains. For more science in your day, subscribe to Seeker. Are you wondering how our brains power our thoughts in the first place now? Don't worry, we've got it here. Did you know?
There, check that out. If you want to learn more about what this man has, I think he did a very wonderful, brief, and succinct presentation. There it is right there. And thank you, sir, for letting us share and promote your little production. We do appreciate that. TMC, comments? Kind of fascinating, isn't it?
Yeah.
It's pretty cool.
I think they know more about the brain than a lot of these people are letting on to.
This was elementary, right? I mean, this is real simple stuff, all right? We know, and we've seen, and we've shown you that there's so much more advancement in it. Like, for example, being able to only image or map 302 neurons. Oh no, we're already way ahead of that. But these guys actually took it all the way. The point about this is not that they were able to map, to a very high degree of granularity, but they were able to extract the thinking of a very simple animal and put it into a machine completely and have that machine be able to steer and run.
Yeah, but it's not, we're taking this particular worm and this particular worm's neural circuits and neural impulses, and we're going to transfer them into this robot and it will come alive. No, it's not. That's not what happened. It's not what happened. they map the neurons and they put it through something like a neural network, artificial neural network, or some type of a learning network. There's many different terms or subsets of what a neural network would look like, but then they just put the sensors on the robot front and back and allow that to be the stimuli that runs over the artificial neural network that is just a copy or a recreation.
That's right.
Like an artist making a painting, or not even necessarily a photograph. It's more like an artist making a painting. Go ahead.
Simulation. You remember that little part he said about, well, he said the other guys put this into a simulator, and I can't pretend to know how that works. But there's so much that you can do by really looking at nature, something natural like a brain, and then getting a really good, exactly as you said, a really good rendition or copy of that, and loading it into a simulator. And but this is before you even get to the point of trying to reverse engineer nature. It's like you just study how that whole thing works. And if you ever played with electronic circuit simulators, I've built circuits both ways. On a breadboard, do wiring things up myself, or just take all of those components and the parameters and my desired output that I want to get for a certain input and just load it into a software simulator. And it's really neat. It's really quick, and you can learn so much. So imagine having a very sophisticated simulator and just how much they could map out what it is that they ultimately want to build.
That's what they're touting with the whole quantum innovation, quantum revolution.
Better computers.
Yeah.
More powerful simulations.
Yeah, modeling materials and medicine and everything else, just to run simulations. The number of simulations.
Yep.
Exponentially goes up with quantum computers. What would take?
Yep.
Yep. I forgot what they said. Thousands of years. Might have been more than that. They could run them in,
I don't know,
seconds.
Man. I don't know.
The computing power went way up.
I'm scanning the comments out in chat from Mr. Dynamex right now, and there are all sorts of little bits and pieces that you can pull from things like Nova documentaries on PBS, even from science fiction movies, and what's in some of those things with the way that they have kind of brought it to the big screen about how to make something synthetic in whether it be a life form or a synthetic reality or a hologram, all this stuff. There are threads that we can draw from all of this. There's another show coming on one of our sister networks, Patriot Soapbox, tonight at midnight, where we'll be getting a little bit more into that. And this science fiction really comes true. It really does. We talk about med beds now, and that's something that they're working on perfecting these things, and with certain frequencies, they can alter the function, morphology, or malfunction and healing of cells in a body. Well, that med bed is something that was on Star Trek 30 years ago. Maybe it takes 30 years, but this stuff comes true. So you want to know where we're going? Perhaps check out today's science fiction. I have another little video if you'd like to see it. These guys are, I think they're also, they have a very lighthearted view of this whole situation. They're making an analogy between neurons, as we were showing them when you had a particular graphic up last week where there was a kind of a matrix of neurons that were up, and they were being connected, if I'm not mistaken, by some sort of nanotubules which helped to guide the way that those neurons interconnected with each other. If I'm not mistaken, that's sort of what it was. It was like basically brain on a chip. So these guys are kind of speculating about that a little bit. So I'll put this one up. I haven't decided how much of it I'll play, but that looks like a friendly guy here. And so anyway, here he goes.
A simple model of a neuron has three parts: the main cell body, dendrites, which receive signals, and axons, which send signals, and can sometimes span an entire limb. One of the things that makes neurons...
So did you catch that? I'll back it up. This isn't something that we covered particularly in our discussions about brain neurons, but let me just back this up.
Special sometimes, which receive signals, and axons, which send signals.
Okay, it is true. Nerve axons can be very, very, very long. And that is something that can go a long way from the originating cell. And I'm not sure whether you knew that or not, TMC, either. But particularly with the skeletal muscles and things that are at great distances, the final end of the axon can be a long way away from where the nerve cell is. And it's just something to think about. You know, they're kind of like wires going through our bodies. And you can get a pinched nerve or a fractured nerve, but what's usually really happening is it isn't the neuron that's getting pinched, it's this axon part that's getting pinched someplace. And nature's very ingenious. When an axon goes a long way, it is covered by, you guessed it, an electrical insulator, which is based on fat, and it's called myelin, and it's called a myelin sheath. And the end of a nerve can be many inches away. I just thought I'd throw that in there.
And can sometimes span an entire limb. One of the things that makes neurons special is their ability to generate and transmit electrical pulses over relatively long distances. And they do that reliably and fast enough to respond to the environment. So Galvani was on the right track?
Wait a minute, this is the same guy. He's got a doppelganger next to him. Very clever, very clever. And you might want to watch more of him too. Fascinating. Yeah, we get pretty deep into the patents and the design and the technical stuff. So it's good to have a little entertainment once in a while. And these guys are bringing facts while keeping it lighthearted with very good visual expressions as well.
Kind of, but we've come a long way in our understanding of electrodynamics since then. Back to the timeline. We'd have to wait until the 20th century before scientists really pinned down the biophysics of this electrical axon thing. In 1906, Ramon received the Nobel Prize for his pioneering work on the general structure and function of the neuron. But it wasn't until 1952 that Alan Hodgkin and Andrew Huxley teamed up to discover the biophysical mechanism for electrical pulses in neurons. Hodgkin and Huxley were both biophysicists, so they decided to tackle the problem. And they did their experiments on the squid giant axon. A giant squid axon? Like 20,000 Leagues Under the Sea kind of giant squid? No, not a giant squid axon, a squid giant axon.
So this is a TMC. You might want to just throw in a little addendum to that idea. It's really handy if we are trying to figure out something about a human body and there's a similar part in a different organism that's really big, or if we can do something to treat those small structures in the cell, the cellular structure or whatever it might be, to make them grow or get larger, to bring them from the microscopic world up almost to the macroscopic world, that also is a lab technique that helps us to study these things. So these guys, these old guys, I think it was H&H, they decided that they wanted to learn more about axons, so they picked a really big animal that happens to have big axons and maybe big nerve cells too, for all I know.
The axon was giant, not the squid. That made it more accessible and easier to work with. I mean, who wants to work with a tiny human neuron, am I right? Anyway, if you look up the Hodgkin-Huxley model, you'll probably see differential equations that look like this. Of course, equations don't exude intuition, so let's try to make a visual instead. Think of a neuron like a leaky bag of charged liquid. It's mostly water, but it has ions like potassium, sodium, and chlorine dissolved in it. A neuron, like all cells, has a two-layered cell membrane. When it's not doing anything, the neuron maintains a potential difference, a voltage, across the membrane. How does it do that? Ion pumps. Ion pumps run on chemical energy known in the bio world as ATP.
So I got to really hand it to these. I almost said these guys, but I think it's one guy, because in a much drier spirit, we kind of covered this also. There are ion pumps. There are ion channels that are semi-permeable based on the conditions, and they will either admit or refuse to admit certain molecules, which will change the charge of that neuron from between the inside and the outside, and which reaches a certain potential, that's when the nerve will fire. And so this is kind of pretty good diagrammatic review here.
This is a major reason our brains consume 20% of the energy we eat. Outside the cell, there's a higher concentration of positive ions like sodium. Inside the cell, there's a higher concentration of positive ions like potassium. Negative ions like chlorine are spread out all over the place. The ion pumps are specialized protein structures embedded in the cell membrane that pump specific ions against the concentration gradient and against the electric potential, if necessary.
Fight the gradient!
I know, this is all sounding very biological, but it's just electrodynamics. It's just circuit stuff. Ion pumps use energy to move electric charges around. That's what batteries and...
It's really pretty simple stuff. It just ends up being that it's at a big scale. You have a lot of these, but at the very simple level, it is really just as he said. You take one nerve cell and you want to have that either be in a dormant, inactive, standby state, then keep the electrical potential at that minus 70 millivolts. It's just sitting there. It's like your computer on standby. And just to review, this is, we're going to have this is perhaps the environment inside the nerve cell, and this is the environment outside the nerve cell. And these pumps, I'm pretty sure, I'm not quite sure, but I'm going to guess that they can pump both ways. Based on the signals that they're getting, they will either allow ions to go from an area of positive charge to negative charge or an area of negative to positive, depending. Not quite certain of that, but it's something along that line. And then you just take this and multiply it by millions and millions and billions of nerve cells and interconnect them all, and that's kind of how it works.
Other voltage sources do. In the Hodgkin-Huxley model, ion pumps are like batteries. Remember how the cell membrane has two layers? Well, that's just a capacitor. The ion pumps built up electric charge on either side, creating a voltage. In the Hodgkin-Huxley model, the cell membrane is a capacitor. Ion pumps are not the only devices that span the cell membrane, though. There are also ion channels. When these channels are open, ions can and will try to balance their concentration.
So as we were covering on, I'm not sure whether it was one or two previous episodes, but we took this way, way, way further ahead from this. This is a basic diagram. But now if you can envision getting an exterior addressable ion channel so that it's possible to control whether that ion channel either admits or refuses those charged ions, now you can externally control whether that neuron fires. And that's kind of what we were talking about last week. And on your previous episode, TMC, you even carried far beyond that in terms of how these modifications are made genetically. You still there?
I'm here, Andy. You're doing just fine. I really, really like this video. I've looked at quite a few that talk about ion channels and ion gates and all that, and this one presents it, I think, in a way that keeps people somewhat interested, because this is stuff that people are like, why do I need to know this? And
the beauty
really
of
do
this,
need to know this. You really, really, really need to know this. This is exactly how your body works, and this is exactly how directed energy systems interact and interface with your nervous system and your brain.
Right. Think of other effects. Exactly. This is a normal operation, but it's so easy to go from this very, very basic and very clear diagram and now add on over here something that is controlling the operation of this ion channel. And that's what we've talked about. So now it's putting a remote control on it. And then, this is just one, but multiply this by 1 million neurons or 1 billion and remote control a whole bunch of them. And now address each one of those. Say, this is one neuron, and we've got a control on it. And now imagine that instead of this one, there's another one next door and a bunch more. So it's really cool.
All hail the gradients! Ions are just electric charges, and when charges flow, we call that an electric current. It's positive charge, too, so the idea of conventional current isn't even a lie this time. But what really matters here is what the ion channels represent in the circuit. The ions don't flow through the channels all the time, and they flow at different rates at different times. That's a lot like a variable resistor, one controlled by some kind of timer. In the Hodgkin-Huxley model, ion channels are variable resistors. They control the electric current by Ohm's law, just like any other resistor. Each ion channel is selective, though. It'll only allow a specific type of ion to pass through.
So I was tinkering with electronics when I was a kid, and the knob, the volume knob on your radio, or on your radio before it was a digital radio and it had an up and down arrow. That was a variable resistor. And between variable resistors and fixed resistors and capacitors, which is what he's equating the membrane of this cell to, that is the major components of any electronic circuit that I built when I was 12 years old. Just add in either vacuum tubes, which I learned on, which are gates. Those are gates that can be controlled, or transistors, which are also just gates, which can be controlled from the outside. And this really, it blends in real closely to old school electronics and modern electronics, which just use way, way, way more gates and way more resistors and way more little capacitance devices. But it's the same idea. It's been made, you just have a whole lot more of those little devices in a much smaller space. And if you consider it, the electronics of life it's almost the same as electronics that I could breadboard on a printed circuit board or actually do it literally on a breadboard. It's just that it's so much smaller and it's so much more efficient. And I mean, it's at the molecular level. There can be so much control and circuitry in an organism that it would have been, so much more primitive to try to compare that to the stuff that I used to build. Anyway, it's the analogies are real close here. So this if you want to know how that human body works at the cellular or at the interneuron connection level, he's really explaining it very well.
Sodium channels only let through sodium ions. Potassium channels only let through potassium ions. So how are they variable? Because they're voltage gated. There are actually several different ways an ion channel could be gated. Chemically gated channels on cells that neurons talk to, mechanically gated channels that are sensitive to pressure or stretch, and even light gated channels in the eye.
So hint, 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 buzzwords that we've talked about all of this in previous episodes. We're talking about chemically inducing channels to open or close, or to do it by way of mechanical vibration, longitudinal waves at high frequencies, perhaps ultrasonic level. Or we've postulated that due to the way that they can target so closely, right down to a pinpoint, that it could be done by coinciding electromagnetic waves so that the interference pattern setup creates a focal point right at a neuron, or maybe even at a part of a neuron. But they let us know that they can get down to about the distance of a millimeter. But I have a feeling that they can get even a little bit more precise than that. But I'm just guessing. Haven't seen that. So we'll let him go on here.
But the ones in the axon are voltage gated. How open they are depends on the voltage across the cell membrane. And that voltage can change with time. In fact, it will change with time as long as that neuron is trying to talk to other cells. These voltage-gated ion channels are vital for getting a signal to the end of the axon. The signal kind of rolls its way down the chain.
It's kind of like a wave. It's kind of like the charge is being passed along, right? So the wave starts. In fact, by the way he's depicting it there, which is most likely accurate, it probably is going in wave after wave after wave until the whole chemical charge balance is satisfied and it goes back to neutral again. So I have a feeling that firing is not just one spike or one pulse or one square wave. It's probably a series of them until they have all dissipated their stimulus.
Hodgkin and Huxley modeled this process as a simple electric circuit. The cell membrane is a capacitor. The ion pumps are rechargeable batteries, and the ion channels are variable resistors. Let's take a look at the voltage across the cell membrane, aka the capacitor, as the nerve tries to send a signal. There are 4 stages: resting, deep.
I'm sorry to keep stopping this, but I know that there's a broad audience that will listen to this. Anybody who's an electronics guy or gal, this is going to seem like, wow, it's so simple. Because you've been playing with all of these components, building whatever it is that you've been building for perhaps a long time. For the uninitiated or the non-electronically minded people, you don't have to understand electronics to know this. It's just to know that you have a capacitor, which is something that can hold a potential and hold it at bay, like a battery. And when you have a resistor, it's like a gate that can be opened a little bit or a lot or closed. And when you have a pump, that's like what, TMC? When you have an ion pump, what's that like?
Man, you lost me. What is it?
Oh, well, that would be like having the force coming out of a battery pushing across a resistor and it wanting to equalize its charge. In other words, positive and negative will neutralize each other. So when you actively pump something, you technically would be actively charging up the battery, right? Or actively discharging the battery. So it's just like a battery that's being charged or being discharged, and doing it at a particular rate based on how many channels are open to allow the charge to equalize or to get charged up or closed off. So it's all very, very much like electronics. Yep. Here he goes.
Polarization, repolarization, and hyperpolarization. Jargon alert, jargon alert. Polarization stages. Resting, depolarization, repolarization.
Okay, so right there, depolarization would be equivalent to discharging your battery. You're using the charge out of the battery to do some form of work. And then repolarization would be recharging the battery. Uh-oh, I lost my audio. Hey, the thing's not going.
And hyperpolarization. Jargon alert, jargon alert. I've been dropping bio words this entire video. Now is the time you get on my case? Okay. I suppose technically the membrane is polarized as long as there's a voltage across it. Which, as you can see, is true almost the entire time. It blows.
And what's a charged battery? A charged battery is something that's got a voltage across it. So your car battery is charged, sitting, waiting for you to start your car, and there's a voltage across it between the negative and the positive terminal. And that, when it's used, when it's allowed to flow from the positive one to the negative one, if that circuit goes through the starter of the car, then that starter starts the engine. And that is ultimately flowing from technically it flows from the negative to the positive, by the way, but that's a small point.
Right past zero, twice. But we have to tell these stages apart. So it helps to think of polarization as just the resting state. The membrane is stable. Depolarization is when the voltage rises towards zero, but overshoots. Repolarization is when the voltage rebounds and drops toward the resting state. Hyperpolarization is when that overshoots. Don't get too hung up on the names. Wasn't this supposed to be about circuits? Oh, yeah.
He's right. Don't get too hung up on the names. But keep in mind, in the back of your mind, why we're talking about all of this? Because we've been talking about how, I'll go back to the diagram here if I can do it easily. Yeah. We've been talking about how, engineered devices can be used to commandeer the control of this. What you're seeing on this waveform would be a representation in a nerve cell of a signal that is being sent. It's being sent down an axon, all right, which is going to result in maybe a finger moving, or it could be something totally different. A signal is being sent down an axon for the purpose of being received by another brain neuron to complete a thought process. So same thing, same waveform, same type of thing, and I'm oversimplifying it a little bit, but it's not losing the general meaning of it, that would either be to move your arm, move your finger, or would be to complete a thought.
It's to feel pain. It would be to regulate your heartbeat, digestion. Tell you when you have to go to the bathroom. Tell your body to breathe while you're sleeping. I mean, this is everything.
Absolutely, it's the return signals too. It's the sensory. It's like feeling something on your finger, and then a signal goes back up to your brain and wherever else. And yeah, exactly right. So and run your heart.
You have a go or you have a no go.
Yep.
And if you have your depolarization, that's your climb up the hill, and then your repolarization is down the hill. If you climb up the hill far enough, then it's going to go all the way no matter what.
Yep.
And so if you cross over a certain threshold on that climb up the hill, then it's going to automatically go all the way to the top, and then it's going to go back down. And what they've been able to do is they can control in real time. They can control where the voltage is. They can regulate that voltage. Right now you see on the screen it's neg 70 or positive 30 millivolts. Well, they can directly interact with neurons, and it can be in a variety of locations in the body, and they can regulate where the new resting membrane voltage is. They can change it, and they can
hold it there too.
Mm-hmm. So they can suppress action potentials from firing, or they can make them all fire, and they can prevent them from resetting.
Right.
So that they're not able to fire again. So you can get them to fire initially, and then you just turn them off. They can stay off if you don't allow the voltages to go back to normal. And if you want to kind of understand a little bit about how that works at a physics level, you look up ion cyclotron resonance. You can look up electron cyclotron resonance. They're kind of the same, but they do different things. But the ion cyclotron resonance has to do with regulating the ions, positive or negative, on each side of a membrane. And in this case, we're talking about a membrane of a cell.
Isn't this beautiful, TMC? Because this is putting into a graphical representation so much of what you've been explaining. And keep in mind, these guys, H&H, Herschel, and I don't know what the other guy, Hodgkins and
Huxley.
And Huxley and Hodgkins.
Yeah.
Those guys worked on this 70 years ago, figuring out what this diagram is. But for all of the intervening years, bringing us right up to the present, there's been more and more of the ability to be able to control these things externally. And this what you've been covering is showing the patents and the technology and the lab work that shows how they've been making that happen. And if we have enough time, we can get into it. We can show you more of what they're doing today. But it's good to really understand the fundamentals, because what you're seeing on this diagram is a completely natural process. This is what your body does just normally. Now imagine what you just said, being able to hold it or being able to depress it or being able to fire it at will based on a target positioned nanoparticle that is intelligently addressed from the outside, whether it opens up those ion channels or not.
Yeah, no, the specific example I was referring to came out of a study from MIT and a school, I think, over in Europe somewhere, England. Anyway, they were doing a study on mice where they were creating what they call temporally interfering electric fields. And what they were able to do is with electrodes on the outside of the head, they were able to regulate and control this stimulation of tissue, where they could prevent excitable tissue from actually being excited, and they could actually excite the tissue, and they could excite it, and then they could keep it suppressed after it excited, all based on how they provided electricity through these electrodes on the outside of the outside of the brain. They could do the same thing with optogenetics. They can do the same thing with mechanogenetics or thermogenetics. It's just different application. It's not exclusive to little pieces of nanotech. It's not exclusive to ultrasound. It's not exclusive to electromagnetics. It's not exclusive to electrodes that are applied to the skin, or any kind of an implantation that we would understand as a deep brain implant or an ECOG array. The real underlying fundamentals is how sophisticated, how well can you regulate exactly what you're looking here at on the screen is this graph or this representation of an action potential.
Yep.
And this is how your body communicates. Your body communicates primarily electrochemically. There are other forms of communication that occur cellularly. The more they understand about quantum mechanics, they're able to measure and understand biological processes under that frame of science.
And now scale it up with really, really powerful computers and the ability to monitor. I mean, just your phone is monitoring it's monitoring more than thousands of parameters coming in and out of its processor, literally millions of times a second. All right. So to think that it's really hard to be able to interface with something as complex as a human body, or a human brain, it isn't that hard anymore.
I think Apple's A11 bionic chip, which is a few generations old, was doing 6 billion processes per second. Billion with a B.
With a B.
Yeah, per second.
Pretty impressive.
That's a lot.
That's pretty impressive.
Yeah, it's not as much as what your body does, but it's a lot.
So this is the basic mechanism. You just take this and multiply it by, let's see, he's going along real well here. And I'll back it up just a few seconds.
Don't get too hung up on the name.
By the way, I see other people joined in the chat. Welcome. Good to see you here.
Polarization is when the voltage rebounds and drops toward the resting state. Hyperpolarization is when that overshoots. Don't get too hung up on the names. Wasn't this supposed to be about circuits? Oh, yeah. Right. We were talking about the Hodgkin-Huxley model. But I think this will make the most sense if we look at everything together. Wasn't this supposed to be about circuits? Oh, yeah. Right. We were talking about the Hodgkin.
Okay, so I'll stop it right on this. So here we are. The capacitor is the membrane of that axon, or that nerve cell, all right? The variable resistor, this is the symbol for a resistor, and it's a variable one, is the ion channel. This is an ion pump. This is an ion pump. So, capacitor, resistor, ion pump, and then a leak. I don't know what the leak is, but maybe they'll tell us.
Leakage, it's normal. The ions go through a normal process of leakage.
That makes sense. Okay, so here he goes.
Huxley model. But I think this will make the most sense if we look at everything together. Let's sync up all three visuals. First, the voltage-gated sodium channels are triggered to open, allowing a rapid flow of sodium ions into the cell. The voltage across the membrane goes from -70 millivolts up to around +30 millivolts. But remember, they're on a timer, So after the initial spike, the sodium channels close. When those close, the potassium channels open, rapidly sending potassium ions out of the cell. The voltage across the membrane drops back down toward the resting state. Finally, after an overshoot, the potassium channels close, and the membrane settles back into the resting state. It does this by leaking a little, and if necessary, by turning on the ion pumps.
Ah, look how that ion pump works. Look at that. That's fascinating. This is an excellent graphical representation.
Yeah, I really like this video.
Yep.
It's a very good video.
All right, let's keep going.
In short, the channels balance the ion concentration, and the pumps unbalance them again. The Hodgkin-Huxley.
So by the pumps unbalancing them again, that is equivalent to recharging the battery.
It's
circuit
normalizing.
successfully.
So, I'm sorry.
It's normalizing the membrane potential. It makes sense. You have two pumps. One does the opposite of the other to put it back
Well,
where it used to be, and it's actually one's working more than the other to that hyper polarization, and then the pump, the one in the middle, is the one that sets it back to the normalized membrane potential.
But define normal. Normal is with a charge.
negative 70 millivolts.
Exactly. So the work of bringing that from plus 30 down to 0 and beyond 0 down to negative 70 is actually recharging the battery so that it will stay in a non-firing state.
And they measure that in ATP.
Yep. Okay, he goes further.
Pumps unbalance them again. The Hodgkin-Huxley circuit successfully models this entire mechanism, but only for a little patch of the cell membrane. This patch only has a few things: one sodium-potassium pump, one potassium channel, one sodium channel, and whatever other miscellaneous devices are around. The signal process starts locally, usually around where the cell body and the axon meet. It then spreads as a chain reaction from one patch of the membrane to the next. Local regions depolarize their neighbors, sending a pulse down the axon. And this happens fast. Fast. The entire spiking process starts and finishes in about 1/1000th of a second.
Okay, that puts a time constant on it, 1/1000th of a second. Fire a neuron in 1/1000th of a second. So if you equate this to the speed of thought, because you're going to have neurons that are proximal to each other. I mean, they're all hopefully inside your head. That means that in a second, you could have had 1,000 of these things fire, and they're probably wired together in such a way. So in 1 second, you get perhaps a complete thought and maybe the beginning of a muscular reaction by something being sent out saying, okay, this is what I see. Oh, finger's getting burnt. Let me pull it away from the stove. Boom. Something like that. Pretty fast.
But how fast does the signal take to get down the axon? Good question. It actually depends. It varies a lot depending on the type of neuron and the animal species. But at top speed, we're looking at about 224 miles per hour, or 100 meters per second. That's the length of an entire football field, including the end zones, in a single second. Compared to how fast signals travel through wires, though, this is actually...
So you can equate this to, and they've done studies whereby a muscle signal that's sent for you to raise your eyebrow on your head does happen faster than the signal that is sent for you to wiggle your tippy toe, because that's further away. And it is measurable, and it does take a little bit more time, but it's not a whole lot of time. It's pretty darn quick.
Pretty slow. Oh no, does that give robots the advantage? Anyway, so it took us a while to figure out neurons. The real work began with Luigi Galvani and dead frog legs, but culminated with Alan Hodgkin and Andrew Huxley and their circuit model. A neuron is like a simple electric circuit, or at least a chain of simple circuits. It's made of capacitors, batteries, and resistors that obey Ohm's law. Components that create a pulsing voltage, all just to tell other cells what to do.
So now if you consider, and we can speculate for just a second, We're talking about analogous components to electrical circuits and that obey Ohm's law. They obey electrical laws that function and are used, exploited all the time to build every kind of circuitry that you use in all of your electronic devices now. So given the level of sophistication that those devices are at now, and given that they operate on, even though they're complex cell phones and smartphones and things like that, they still operate at the root based on very simple principles. How hard do you think it really would be to be able to start to blend the two together? Just generally speaking, not getting into the nitty-gritty of what we've shown about how they do this with purpose-designed nanoparticles for controlling some of these channels. How hard do you think it really would be? Not that hard. Now, the material science and how to actually get, how to perform that and make it work right, well, there's a lot of research that's gone into that, but they've made tremendous progress. Check out some of the previous episodes. We get into this in great specificity. And for more reading, TMC, a shameless plug for humaninternet.org, wouldn't be a bad idea either, because you got some really good stuff up there. So anyway, that's scrolling across the bottom of the screen. And TMC's got a tremendous repository of information there. So he's wrapping up. He's getting to his summary here of all this now.
Let's just hang a simple electric circuit, or at least a chain of simple circuits. It's made of capacitors, batteries, and resistors that obey Ohm's law. Components that create a pulsing voltage, all just to tell other cells what to do. Let's just hang here for a bit and take it all in. So, got any questions about this analogy? Please ask in the comments. Thanks for liking and sharing this video. Don't forget to subscribe if you'd like to keep up with us. And until next time, remember, it's okay to be a little crazy.
So, I think that was a stupendous production, and you will find the link to this video below in our comments section. And I must confess, I do not know this man's name, but I do know that he resides in the Science Asylum. So what do you think of that?
I think that was very good. It's a very important thing to understand, how a neuron communicates. And that's just the electrical piece. There's the chemical piece as well.
Yes. And they're related. They're related.
Very much so, yeah.
Yep. Because different chemical components have different electrical charges, just as a natural characteristic of the chemical component that they are.
Yep. It was a very good video describing ion channel, ion gate flow.
There are two things that you can do. If you want to chemically alter a molecule, you can do it electrically. And if you want to electrically alter a chemical, you can do it chemically. Electrically alter a molecule, you can do it chemically. So it can be done both ways, depending on what is available in the environment where that cellular structure is. If it's more advantageous to alter the chemistry, well, that's going to change its electrical characteristics. If it's more advantageous to modify it electric or to input into it electrically, that's going to change its chemical properties. Starts to get simple after it gets complicated. And then it gets complicated again, then it gets simple again. So.
What else you got?
I got some interesting stuff. It's mostly pretty lighthearted stuff, but I always want to have people keeping in the back of their mind the dark side. The dark side is, oh boy, what if with all of this network of Wi-Fi and 5G and these nanoparticles that we've shown you that they've designed, what if those things can be controlled? What if those little nanomolecules and very, very ingeniously designed little particles are in our bodies and we don't know it? And what if There is very, very sophisticated software control of the either the ultrasonic or the infrared or the electromagnetic environment around us. Just what could those interactions be? And the implications of this are huge, both for therapeutic purposes as well as for other purposes. But we do not seek to engender fear or paranoia, and this show is for entertainment purposes only. I do have another one, and I've got it right here. This one's actually quite short, and I'm going to put it up right now. And once again, the hyperlink to this is listed in the below the comments section in our show notes. And here it is. And here she goes.
The future is now. We've got the entire World Wide Web in our pockets. Chefs have created veggie burgers that bleed like real meat. And scientists can control minds.
You like that?
I think I've seen this before, once upon a time.
I mean, what more could you want than a veggie burger that exudes juice? Okay. But she looks a little bit troubled right now. I don't know why. This is from the channel Neurotransmissions, and here she goes. It's very short.
Wait, what? Yeah, that's not a joke. Hey there. I'm Ali Astrocyte, and on this episode of Neurotransmissions, we're going to talk about optogenetics, controlling the brain using light.
Yes. Sound familiar? That's what we've been talking about for the last couple of weeks on Cyberhuman Frontiers and on Human Internet Late Night with TMC on Thursday nights or Friday. Wait a minute. Wednesday nights or Thursday, depending on what your time zone is.
Yeah, it's Thursday, Friday morning, East Coast time, and then Monday morning, East Coast time.
Fun stuff. Fun stuff, guys. Okay, here we go. Neurotransmissions, take it away.
When I say mind control, it might sound like something out of Star Wars, but in the last decade, advances in technology have let scientists figure out how to directly control the signaling between neurons. See, neurons use electricity to send signals. All along both sides of the cell membrane are electrically charged ions, which create an electrical gradient. To send a signal...
So this is just a review now, because you've already seen the real nuts and bolts of it, but this is very quick. So this just helps drive the point home from another point of view.
Called an action potential, ion channels have to open to allow ions to flow across the plasma membrane, shifting the balance of the electrical charge. This change in the charge sends the action potential shooting down the axon of the neuron, passing the signal along the length of the cell. Since that action potential is the key to how neurons communicate, neuroscientists have been trying to figure out how to control it. Control action potential and you control the brain. Kinda. But it wasn't...
Okay. Sounds good. Kinda.
It wasn't until the early 2000s that they found the right tools for the job. In 2003, some scientists discovered a protein called channel rhodopsin, a light-sensitive ion channel produced by an algae. This channel opens in response.
So, TMC, there we go. Is this what we were talking about 3 weeks ago? Channel rhodopsins.
Yeah, opsins. Optogenetics, you're looking at opsins. Channel rhodopsin is a type of opsin. But yep, this is where they can go in and genetically modify a brain cell or a neuron and get it to respond, open and close those ion channels.
Exactly. Design a molecule and then get it placed where you need it to be placed to get the desired result.
Right.
Yep.
Response to light, allowing the algae to sense the direction of a light source, like the sun. Scientists immediately realized that this new protein could be the answer to their conundrum. In 2004, two separate groups of neuroscientists were able to use viruses to insert the channel rhodopsin gene into neurons. Use
viruses to insert the channel rhodopsin gene into the location desired. So keep in mind, now, if you couple this together with micro-sized lasers that are also situated in optimal locations, and if you can power those micro-sized lasers, which we have, well, I don't have any personally, maybe I do and I don't know it, then it's you couple this together with the channel rhodopsins, and now you've got something which is now electrically addressable from the outside to be able to cause those channel rhodopsins to alter the conductivity and the firing of neurons. And there you go. Presto.
Channel rhodopsin can be activated by shining a light on it. When it's expressed in neurons, shining a light on those neurons opens up the channels, generating an action potential. Scientists have been able to create mice that naturally express the protein in their neurons. And researchers can surgically insert tiny LED lights or optic fibers into different brain regions to focus their light beam on specific structures or cells.
Oh, doesn't that look cute? I like those whiskers. They're just kind of cute. His eyes look bloodshot, but I don't know. Maybe he's had a hard day in the lab.
Cell types. When they turn on the light, they essentially turn on the neurons exposed to the light. This lets scientists examine how turning those cells on and off affect a lot of different things, including controlling the behavior of an awake animal. And because light is so easy to turn on and off, optogenetics allows very precise control of neuronal activation.
Okay, the light is so easy to turn on and off. Yeah, and we can do that. We've got such amazing stuff. I mean, in your inkjet printer, we've got precision. Well, in your laser printer, we've got precision and extremely tiny light transmitters and light receivers. It's like we got all the stuff. Now all we got to do is get it into organisms without killing the organism or without being too invasive. Now, what you're seeing with the mouse is a little bit clunky, but most of that could be internalized, and they're working on that.
Like in the realm of milliseconds. Because optogenetics only requires a single gene to create the protein, scientists can target specific types of neurons to make them express channel rhodopsin.
Okay. Is it all going in? I got to see what the chat is saying here. Let the light shine, and then, oh, geez. Fake burgers. Oh, you guys. Okay. I'm with you. We're paying attention.
Letting them pick apart the roles of different cells in the brain. Even more recently, researchers have started using new light-sensitive proteins to influence neuronal signaling. Some of them open and close at different rates, affecting the speed of the neuron's signal. And some of them can actually suppress action potentials, letting scientists effectively turn off particular neurons within a circuit.
You're on mute. Indy, you're on mute.
I'm muted. Thank you. Okay. Thank you. I didn't realize that. Take it to the next step now. Let's say that we have got these micro-sized emitters, and now we have designed nano-sized particles with channel rhodopsins on them, which are sensitive to different wavelengths of light, different colors of light. And if we've got emitters which are capable of emitting in different wavelengths of light, now in the same size package, we can start getting more functionality. And guess what? They got all that stuff too. She goes on.
Using optogenetics, neuroscientists have been able to discover some pretty fascinating new stuff. Some researchers have identified a circuit in the amygdala, sometimes thought of as being the fear center of the brain, that's involved in how we learn to fear something. And they've also been able to tease apart some of the circuits that are affected in movement disorders like Parkinson's disease. Other researchers, including some of my friends here at UCSD, are using optogenetics to understand things like how the brain processes time and how different brain cells are connected to aggressive behaviors. Some scientists are even trying to use optogenetics to help human patients by developing new kinds of pacemakers that use light to regulate the heartbeat. That's the beauty of a technology like optogenetics. It can be used for so many different things. The possibilities are really endless. I guess we'd better be careful and make sure we keep this technology away from the mad scientists, huh?
What do you think? There are mad scientists out there, aren't there?
She kind of looks like a mad scientist to me.
Was it what? I don't know. Did she looks very pleasant to me? Maybe looking a little to the right.
Light to regulate the heartbeat. That's the beauty of a technology like optogenetics. It can be used for so many different things. The possibilities are really endless. I guess we'd better be careful and make sure we keep this technology away from the mad scientists, huh? Thanks for watching this episode of Neurotransmissions. If you liked it, give us a thumbs up and hit subscribe to learn more about the new kinds of technologies scientists are using to study the brain. Until our next transmission, I'm Allie Astrocyte. Over and out.
Well, that's wonderful. Allie Astrocyte. You know, that's good. You're going to witness me give her a thumbs up right now. Look at that. Good job. And hopefully she's not a mad scientist.
Yeah, I was just kidding.
You want to dig into some patents, or would you like to continue with more of this? Stuff.
I think this is good. This broad overview is very important because this is.
It's true.
Wow.
Because
how do you frame all this stuff when you look at how are they going to plug life into the internet?
Right.
How are they going to connect life to the internet so that it can be understood and monitored and managed and manipulated, basically?
Look at the nexus of 5G technology, increased artificial intelligence capacities,
material science, biotechnology, synthetic biology,
increased permeation of our environment with intelligent electromagnetic signals, increased technological facility to design viruses as delivery packages to deliver these types of little, nano-sized devices to the specific location where they are desired to be. You heard what she said. Get that particular designed little nano device right to the specific type of neuron that you want to get it to. So just put all the pieces together. There it is. It's totally cool. Totally cool.
Yeah. What's the next one, Indy? What else you got?
Let's see. What have I got next?
You got 45 minutes, and
I got, this one's only 6 minutes long. Got another one here. The title of this is, Did We Just Figure Out How to Program a Brain? And this one comes from SciShow. So I can put this up. And it really is we really want to ask you to just in your mind just put all of these little pieces together like a puzzle and say, wow, yeah, they can do it. And then look at the world around us. Look at what's out there. Okay, look at gene therapy. Okay, which a couple of companies, I don't want to start mentioning names or getting into anything. Who knows? God knows. You know, the streaming platform might take our video down. But look at applications of gene therapy and transfection, which is the process of utilizing an RNA, a messenger RNA string of data. Basically, it's a piece of genetic code, and getting that into a cell and into that cell's processes. Look at that. Look at how that can be delivered, and look at what can be delivered. And look at what you could even reprogram that cell to build on its own. So yeah, stuff like that. Let me put this up. And where did it go? Here it is. Okay. Did we just figure out How to program a brain? Okay, so this is SciShow Psych. And we're going to start this right out here. And here he goes. And thank you very much, SciShow Psych, for allowing us to show your little presentation. It's only 6 minutes long, but I'm sure that it's information-packed. Take it away.
I hear people make comparisons all the time about how your brain is like one big, ultra-sophisticated computer, and that's fine, but the idea really only goes so far. Because, like, you can't really program your brain like you can your desktop, which is kind of a bummer. I want to know Kung Fu, and if we could just get in there and start manipulating neurons, we could solve a lot of the struggles our brains and bodies cause. But what if we could?
But do you remember about 8 episodes ago when we had a couple of people connected by way of electronic interface devices, and there was a third person there who was utilizing something that seemed only like it was a tablet. It didn't seem like it was an extremely powerful device to use to transfer the thought information from one person to the muscular operation of another person. We showed that. And that's, you know, that's just the beginning. Now take that and make it more powerful and better. And we have also featured an example of a person. I don't know whether it was, I'm not quite recalling what particular type of specialized muscle learned activity that it was. But the concept was to give someone an ability to do a complex muscular task without ever having to practice it for, you know, some number of years to gain mastery over it, just by getting those particular signals into the right part of their brain. So it's kind of the direction that he's going in now, and he's really just substantiating and giving credence to what we've already shown you. So there's lots of good stuff to review.
Want to know Kung Fu? And if we could just get in there and start manipulating neurons, we could solve a lot of the struggles our brains and bodies cause. But what if we could do that? Recently, scientists working with mice have been able to send new signals into their brains, ones that seemed so natural, the mice may have no idea they weren't their own. It seems a little sci-fi, but it's real research, and someday it could lead to some pretty amazing new treatments. These studies center around.
Well, would it be amazing if I was doing something and I didn't know that it wasn't me that wanted it done? That would be pretty amazing if I didn't know that I'm the one that wanted to do it. Because I thought I wanted to do it, but I didn't, but someone else did or something else did. I mean, I'm just speculating for entertainment purposes.
A branch of neuroscientific research called optogenetics, which makes use of light-sensitive proteins inserted into neurons through genetic manipulation. Once the proteins are there, researchers can control the firing of those neurons using just a flash of light. We actually did.
So you see what he's saying? It's just we're saying the same thing over again. Got to get them there. And I think we kind of got into this a little bit more last week. You said there's 2 parts of this. One is actually 3 parts. You got to design the right nanoparticle, and then you've got to get it where you need it. And then once you've got it where you need it, then you have to target address it to get the desired function out of the person. Oh, excuse me, the mouse that you want to get it out of. Okay.
A whole episode on the mechanics and history of optogenetics that we'll link to after this. But the general idea is that this method alters what certain receptors on cells respond to. Instead of waiting for a chemical neurotransmitter to tell them to do their job, an altered receptor will take the command from a flash of light transmitted through a wire. When optogenetics first entered the scientific literature in 2005, the initial technique involved flooding a relatively large area of the brain with light to activate it. This is still generally the way it's done for now, and it does work. With it, we've been able to get groups of neurons to fire and even induce behavioral changes in rodents. Like one experiment used optogenetics to get rats to walk in circles, which is pretty amazing. Also a little upsetting.
Why is it that I have to do this now? This is my life. I'm upsetting. Why is it that I have to do this now? This is my life. I'm a rat.
But one of the downsides of this method is that lighting up a whole area can be imprecise. Trying to tease apart what's happening with the specific neurons you want to study versus what's coming from ones lit up in the crossfire can be pretty darn tricky.
But I got news for you, or TMC has news for you. It's not going to be necessary pretty soon to actually use wires or, fiber optic fibers, which are basically thin pieces of glass. It's going to be possible to build structures or to command the organism to build those structures. Think of them as very small optical scaffolds, which can be, which the cells can be programmed to build on their own. Okay, so you don't have to put them in from the outside.
Yeah, when you're talking about growing bioluminescent components into cells, where they genetically program cells to emit particular wavelengths of light, what they would call bioluminescence.
Exactly.
Mm-hmm.
And is this not
have
possible?
And then have adjacent cells that are responsive
Receptors.
to bioluminescence. So you'll have the ability to monitor and measure the bioluminescence with the use of light scattering through the body. And you'll be able to potentially, in my view, use bioluminescence as a stimulation technique if you can get enough sensitivity built into the receptors.
Right. So now I ask you a question. Does the abdomen of a firefly not light up?
I don't know. You set your farts on fire? I'm kidding.
Well, if the tree falls in the woods, did the firefly fart? No, I don't know. Okay.
The firefly lights up, buddy.
If this was something totally far-fetched and totally sci-fi, then imagine that we lived in a world where there were no bugs or no squid or no jellyfish that ever emitted light. And the only way that they did is because we, as mad scientists in a lab, created this unusual chemical and we injected it into them and we gave them this new functionality. But wait, no, they have this capacity for bioluminescence naturally. And that means that there is genetic code that causes those cells in those fireflies to turn their energy, the calories that they eat, into light. They are organic light emitters. So if a certain string of genetic code is able to cause cells to create compounds that can light, then why can't we just program other cells in other organisms that couldn't previously have that bioluminescence to bioilluminate? And why couldn't we tell those bioluminescent molecules where to go? We could. It's all there, all of it. This piece is there too. So that light that he's talking about shining in from the outside, no, we're already we're on the verge right now, if they haven't already done it, of bringing those microscopic pinpoint light sources on board in the body and are going to be powered by the energetic processes right within the body. You won't even need batteries to run this. Okay? It's all there. It's already there. Guys, it's a brave new world. We get so excited. Are you as excited as I am?
I'm horrified and petrified, and I'm amazed, and I admire the beauty of it. I mean, it really is what is that book? A Tale of Two Cities, something like that. Best of times, worst of times.
Something like that.
Yeah, it's really what.
So let's take it further.
What it is.
Let's take it further. Fireflies are really boring, right? I mean, they just light up this yellow green. It's boring. It's just one color. I mean, wouldn't you like to see like blue, purple, kind of like a Christmas tree with all different lights on it? Wouldn't you like to see fireflies that lit up in different colors for more fun, for more variety? Well, guess what? There are creatures who can completely change the reflectivity and the color of their skin in split seconds. There's a kind of squid that can do this. So I mean, fireflies are just not that advanced. We could easily go in now and reprogram fireflies to light up in any color that you want. All right? We could do that. Maybe somebody's already done it. Anyway, I'm digressing way too much. Let's go. Let's let this gentleman finish.
However, the times, they are a changin'. Thanks to advances in tech and biology, a group of scientists recently figured out a way to target specific individual neurons rather than a relatively large patch of tissue. They published their results in April 2018 in the journal Nature. And brace yourself for some sci-fi, because they're doing it using holograms. Visual holograms, the kind you see in Star Trek or Star Wars, use multiple beams of light to project an image. But holography in general is really just about light interference, and that's what this technique relies on. The team called their method 3D SHOT, which stands for three-dimensional scanless holographic optogenetics with temporal focusing, which I'm glad they have such a good acronym for that. Unlike the original optogenetics technique, 3D SHOT focuses light from several sources on individual target neurons. Only the specific neurons in the tiny area all the light crosses.
Holy
Did we
shit.
just say this?
Do you have, Andy, did you
I
send
can't
this
hear
video-
you.
Did you
Oh,
send this video to
could
me,
you
buddy?
speak louder, TMC? I'm not quite hearing you.
Did you send this video to me?
Yes.
Holy shit. That's
Of course.
amazing. That's exactly what the laser systems do in your cell phone.
That's right. I send
when
you.
they're
I
talking
send
about-
you
when
everything I find, just like you send me everything you find.
When they're talking about the multiple beams of light that intersect, creating basically a holography, not a hologram as we understand in the movies, but holography with respect to an image where they look at light interference. It's exactly the way that the dot matrix, so to speak, works on your Face ID when you log into your phone, and for 3D mapping for augmented reality functions.
Precisely.
They're saying that this stuff. Wow. Anyway.
Yep.
That's amazing. Amazing.
Well, it's kind of funny because we dig and do research, and we kind of put the pieces together, and then someone else comes along and says, "Yeah, here it is," and we say, "Oh yeah, gee, this is what we were scratching our head about like 5 weeks ago," saying, "Yeah, yep, here it is."
Is receive enough light to activate. Light from several sources, optogenetics with temporal focusing, which I'm glad they have such a good acronym for that. Unlike the original optogenetics technique, 3D SHOT focuses light from several sources on individual target neurons. Only the specific neurons in the tiny area all the light crosses receive enough light to activate. That can eliminate a lot of unintended firing, so these results are much less muddy. And it seems effective. Scientists using this method have already observed mice brains firing at a natural timing and frequency when stimulated. They can tell based on the amount of calcium the cells are using, which is a good estimate for the rate of neural firing. And because that firing appears to really closely mimic the way their brains would naturally detect and process stimuli, it's likely that to those mice, any experiences induced using 3D SHOT would be pretty realistic. I mean, unfortunately, we cannot ask the mice about this.
Oh, there's a mouse with a microphone. Well, wait a minute.
We need to put
If you could just understand mouse language.
a woke on top of that mouse. Just put woke right there.
Yeah.
Yeah. Put a syringe right up its ass.
Oh no. Come on, this is a family show.
Yeah, you're right. For entertainment purposes only.
So, and it's
Don't forget your
entertainment.
booster. Don't forget the booster every 6 months.
No,
ladies and gentlemen.
no, don't go there. All right. So the deal is there's one little takeaway I wanted to bring, which was that he specified that there can be literally a pinpoint location where there is sufficient energy coming from multiple sources so that is the exact, precise, and only target neuron that will be caused to fire. And that's brilliant.
Wow.
It's brilliant. So let him go. He's more than half done, and we have less than half of our time left. Okay, here he goes.
but if we can stimulate their brains into firing in the same patterns as it would when, say, seeing, feeling, or smelling something real, that seems reasonable. One way we can test this hypothesis is by observing actual behavioral changes, like a flick of a switch causing a mouse to stop in its tracks, which we haven't done yet. But the good news is those experiments are currently underway, so we'll keep you updated. If scientists are able to make neurons fire realistically, though, and if those experiences feel real even though they're not, well, in theory, this technology could eventually be used.
I'm just thinking, my imagination goes crazy, all right? So I was just thinking, and I don't know why I was thinking of ballet, but imagine the most graceful ballerina or male accompanying ballet guy that was doing this. Imagine if you could take that muscle memory and those patterns of muscle firing and dump them into a mouse, and you could get a mouse to dance to the Blue Danube Waltz, or do you imagine what you could do, and the mouse would think it did it, which it did, right? Imagine that. All you got to do is capture those patterns, those neuronal firing patterns from the person who has the skill, and then translate them to the appropriate muscle triggering centers of the mouse. And, it's not going to maybe do it quite as gracefully, because mice tend to be a little fat and a little bottom heavy. But apart from tipping over once in a while, you could literally get a mouse to do a ballet. Just saying.
Used to alter memories by editing the way neurons associated with those events fire. We could make mice perceive things that aren't really there, all with the flash of a few lights. We could download kung fu into their brain, like the Matrix.
Oh, he just said it, not kung fu. Why is he going to kung fu? I'm going with something beautiful and lovely and an artistic expression. And he's going all kung fu.
Kung fu is all of those things.
All right. I give up. Okay.
It is.
It's got some art, skill, and grace to it. Yep, absolutely. Mm-hmm. Okay, so it's true. It's true.
Everything about it's beautiful except the violent stop that occurs rapidly with every punch or kick. Everything else up to that point of the violent stop is beautiful.
And this stuff is all beautiful as long as at the end of the day, it doesn't result in a violation of the will of the subject target. So in the case of the mouse, if that mouse had been watching television a lot and it was always watching the dancing channels, and it said, man, I wish I could do that. I wish I could do it. And then if it could do it, that would be one of the happiest mice that ever lived. Okay. But if the mouse didn't want to do that, or kung fu, or whatever it might be, if the mouse didn't want to do it and it was somehow induced to be forced to do that, then that would not be a happy mouse. It might even be an angry mouse. And you don't want to mess with that.
You're not looking at it sophisticated enough in this fairy tale. If technology is sophisticated enough to force a mouse to do kung fu, why wouldn't the very same technology with that level of sophistication just override the mouse's emotional spectrum and make the mouse feel blissful all the time?
It could. Absolutely. Yep. I just didn't go there. Yep.
Psychotronic, positronic, managed, psycho-civilized society.
Yep. You could make that's absolutely.
Musk said it. The first person to the limbic system wins.
He did.
That's where the pleasure center is. That's where the reward center is. That's where the fear center is. All of it. The limbic system, primary component of the emotional experience that is the human organism.
Now, I think that this gentleman knows this stuff, and I think he has just about he's got a similar. His imagination is on par with ours. All right, and I think he looks at all these possibilities. So I'd love to talk to this guy, SciShow Psych, Once again, the link to the rest of this video is down
It's
there.
very, very good.
Anyway, he's almost finished.
Rats doing kung fu. That things that aren't really there, all with the flash of a few lights. We could download kung fu into their brain, like the Matrix. Finally, rats doing kung fu. That might be getting a little ahead of ourselves, but honestly, maybe not as far as you'd think. The research team responsible for 3D Shot already have a few ideas about how they're going to sort of copy and paste brain activity to achieve something like this. If they can measure the sequences of activations in specific neurons involved in the behavior they want to program, 3D Shot could feasibly send that pattern into a different brain. Basically, programming it. Brain programming. Now, even if we're making great progress with mice, applications in humans are likely a very long way off. Optogenetic methods require specific light.
TMC, how far off do you think, really?
Well, considering that they're publishing papers on it that are now out in the public, it's probably already been done, man.
Exactly.
I mean, it's just
it's
ignorant.
not far off.
It's totally ignorant to think that this technology is going to be displayed or even discussed in public first.
And I appreciate I appreciate the levity that SciShow Psych brings to this, but to some extent, and I'm sure it's not intentional, it's almost a way of normalizing this type of impending technological encroachment on people by delivering it with. He's not doing it on purpose. He's excited about this stuff, but it serves to normalize some things that we might start to see in the world around us, and we're just, it's not going to be any big deal. It's done. I mean, it's been done. I won't even say it, but with certain things that have happened in the recent past. With a certain unexpected, unscheduled little microbe that got released. A lot of things seem to have become normalized overnight, and people seem to accept it without any problem. And I paraphrase grossly there. But anyway, he's going to finish up now.
Sensitive proteins to be placed into the genome, which could get risky. But if we could do it safely, that would be hugely useful, not only for research, it would be for research, but also for future treatment options. Think about it. If we could design a
treatment options, what is that? Oh, it's an eyeball. Future treatment options. Okay. It'd be great.
Prosthetic eye that picked up visuals from the world and transformed them into the right patterns of neural firing, you might be able to restore someone's vision. That's just one out of thousands of examples, but we have a lot of work to do before we can get there. So I'm sorry if you wanted to download very good dancing skills. I also want that to happen to me, but it's...
I swear, I didn't hear him. I didn't preview this whole video. He's saying what I was thinking, and he said it. Never mind.
It's going to be a while. Until then, maybe we should take some classes until the research is a little further along. Thank you for watching this episode of SciShow Psych. If you'd like to learn more about how optogenetics works and how we figured it all out, you can watch our episode all about it over at youtube.com/scishow. And as always, don't forget to go to youtube.com/scishow psych and subscribe.
You know what? I'm going to give him one. I'm going to give him one. There. I just gave him a like. So there you go. This is SciShow Psych. And I got to tell you, if you consider all of the actual research that we have showed, all of the advances in material science that we have showed you, from nano-sized self-assembling scaffolds of microscopic sized fabrics or tubes or little grid works, all of this stuff, you put it all together and you consider all of these advances. We're not even showing you future. We're showing you present, and TMC brings faithfully all the time the actual patents and documented research and who's doing it, and what the frontier that they're finding that they're at is, and what they've already done. So let's see. Stefan Chin, Caitlin Hoffmeister, Emma Barrett. Yeah, well, the link is down there if you guys want to save that and watch it again or something. So anyway, I also had another one which hey, TMC, you still there?
I'm here. Yeah, man, I'm here.
Our stream is unavailable.
I see that.
Out on YouTube, our stream is unavailable. It was suspended for policy violations right now, at this moment. So what I would suggest that we do is continue on and wrap up the show, and do some conclusions, and then we will we'll upload it. We'll upload it so that people can see it again. Okay? So, summary comments.
Looks like it's back. Go ahead, keep going. Summary comments.
Okay. I think that it's a wonderful frontier that we're on the verge of, and the possibilities and the implications are amazing, absolutely amazing. We did yes, everybody out in the chat, we had a little glitch there, and that's good. I'm just going to say that I think that there are very gifted and very creative engineers and designers. It's not just a hardcore engineering process where it's pages and pages of math, all right? It's also the creative people, the visionaries who help to see a future. They envision a future. And I think it's all in the hands of the future that people envision. And I don't know. I think it really wouldn't be hard for us to speculate for a while. I really would like to engage the chat in the discussion a little bit. What we're going to do now, everybody out in the chat with us, is try to tie this together into what we all and collectively would feel is the best application and the best direction and thrust of these technologies that are literally they're right there for us. Oh, I had another thing too, which I was going to cover, and I won't show it because we're short on time. But there's a group of people who are working on growing plants that have been genetically modified so that they grow meat. And so what you're getting is they're working with grapes, okay? And they're reprogramming the grape so that instead of it growing juice and, that stuff that you make wine out of, it grows meat inside the grape. So you can literally go over to a tree and yank off a few meat berries and eat them. And you're eating meat off a tree. So it's just there's so many possibilities. We're on the precipice of absolutely, utterly stupendous and amazing times. Please, everybody, give us some comments out there. We're with you. We were with you, then we weren't with you. Now we're with you again. And so Val, good one. TMC, what do you think about that? The idea of meat berries on a grape tree?
No, no thanks.
You don't want to
No.
have some sauteed?
No.
In some butter?
No, man, you just make a meatball. Calampolo, for those of you that know Middle Eastern cuisine.
Well,
Really
it's efficient, though. I mean, I'm just going to try to antagonize you a little bit. It'd be a whole lot easier, all right? I mean, if you have to get up in the morning and you got to take and get dressed and go and start the car and drive over to the drive-through to get a bacon burger or what are the Baconator or something, But if you have a little tree right in your house maybe even next to your bed, and you can just lean over when the alarm clock goes off and just yank off a few meat berries right off the tree and just chuck them down. I mean, it's so much more efficient, so much more convenient.
No, don't ever try to use it as a pickup line in a bar either, buddy. I don't think it's going to work.
And so imagine that if it's converting, I'm not even listening to, if it's converting sunlight directly into meat, as opposed to a farmer who's got to go out there and he's got to grow a whole bunch of non-GMO corn and then raise that and then dry it and grind it up into grain to feed to the steers and the bulls out there. And he's got to keep the fences up so the bulls don't run away. And every once in a while, you know, somebody gets kicked by one of them. This guy's walking around on crutches because the bull was in a bad mood and kicked him. I mean, it's so much easier with a tree that grows meat. I'm telling you.
I would want a tree that grew meat like chili peppers, not grapes, buddy.
Okay. Well, all right. I'll tell you what. I think we've done it. We've done it in, and we almost got done in tonight. I would like to just ask you if you would like to gracefully and kindly take us out for the day, unless you have any other comments. Where do you think we might want to go next week?
I don't know. I gotta give you a lot of love, man, because you picked some really good generalized representations of what we've been trying to kind of cover the last few weeks collectively. So it was very good. I greatly appreciate what you brought today, man. I needed that. So
We got you covered. Yeah. Dude, I wouldn't even be here doing this if it wasn't for you. We are a team, okay? Yeah. And we have a vision and a passion, and we see the value and the importance, and so I wouldn't be here without you. So anyway, take us out, pal.
I'm with you. I'm going to diverge away from you when it comes to growing meat on trees, but other than that, I think we're pretty much on the same page. I don't know where we're going next week, but something tells me.
You want to give us a clue? You want to give us a?
I don't know. I think we should stay somewhere in the realm of nanotech and genetics. I mean, this is a really big deal. What's happening right now? So I think we should kind of stay in this wheelhouse because it's a calm, quiet subject right now. Not a lot of people are talking about it anymore. And
Agreed.
We're moving into a new space where genetics and the whole idea of nanoscience, nanotech, nanoscience is really like blossoming right now, and it's something that we know very little about. So I think we should kind of stay in this realm for a little while.
All right. Okay. Well, all right then. Thanks, everybody.
See you soon.
Tune in next time.